Sounds good. All right. Good afternoon, everyone. Thank you so much for joining us for today's Pulmonary and Critical Care Grand Rounds. I'm delighted to introduce our speaker today, Dr. Nuala Meyer. So Dr. Meyer actually attended Yale as an undergraduate. Welcome back, Nuala. And then attended the University of Chicago for medical school. She completed her internal medicine residency and a pulmonary and critical care fellowship at the University of Chicago before joining the faculty at Penn in 2010, where she completed an MS in translational research in 2011. And this year, Dr. Meyer was promoted to Professor of Medicine in Pulmonary Allergy and Critical Care at Penn. So Dr. Meyer works to identify genetic and molecular risk factors for ARDS and organ dysfunction during sepsis to better understand the pathophysiology of these conditions and to seek novel treatments, with the ultimate goal being to develop personalized therapy for sepsis and ARDS. So Dr. Meyer is PI of an R35 on this topic, and she's also multiple PI of the Penn site of the NHLBI-funded APS consortium. She's also the chair of the Critical Care Assembly Program Committee for the ATS 2025 International Conference. And on a personal note, I had a wonderful time working with Dr. Meyer last year. So today she'll be talking to us about the search for treatable traits in sepsis and ARDS. Welcome.
Thank you so much, and I'm really just delighted to be here. New Haven has changed quite a bit in the over 20 years, so it's so nice to be back. Sorry, skipped ahead a bit there. Okay, here we go. Here's the CME code. I do have a few disclosures, none of which I think are terribly relevant to the science we'll be discussing today. And so some of you may be familiar with this really elegantly written perspective piece by David Maslov and many colleagues, but we may be on the cusp of maybe a paradigm shift in critical care medicine. For the past several decades, we've worked really hard to define many different syndromes, and we have very clear criteria by which we could identify patients with sepsis, and you and I would call the same person meeting sepsis criteria or meeting ARDS criteria. And yet, all of this work, along with really important translational and mechanistic work, has failed to give us new therapies for these syndromes that we see so often. And so maybe what we need to do is shift our perspective and stop thinking along the syndromic axis, but rather think about traits that might be common to pancreatitis and sepsis and ARDS. But if a patient, regardless of insult, has that one particular trait, and if we can identify it and be confident that we have a marker to identify this trait, and more importantly, if we have a treatment that we know targets this trait, or a treatment that's harmful if you express that trait, we might modify our therapy and improve the health of patients with critical illness. So what I hope to leave you with is maybe we can start to find these traits in sepsis and ARDS. And I always feel a little intimidated to talk about reactive airway disease at a place like Yale that's developed so much of the science. But I think our colleagues in reactive airway disease are way ahead of us in this field. And I really liked this figure from a COPD paper by McDonald and colleagues, but really trying to dissect COPD into different traits. So underweight, anxiety, vocal cord dysfunction, obstructive sleep apnea, eosinophilic inflammation, each of these would have a different approach. So can we find those traits that are clinically relevant, measurable, and most importantly, treatable? So that will really be kind of the objectives of this talk. We'll talk about this treatable trait framework, think about areas of success and other complex traits and what lessons that can give us for our critical care syndromes. Think about the barriers that are preventing us from having already solved these problems. And then we'll try to shift into kind of our ICU syndromes and think about where we might have had some of these successes and where we could keep developing more. So what are we looking for? This is a bit of a needle in a haystack, and I'm a maybe a simplistic thinker, but I think on the one hand, we really need to find those driver mutations if we use kind of our cancer colleagues as a guide. What's causing the problem? Maybe if we're not going to call it a driver mutation, are there specific causal influencers that if we knew they were causal, then of course targeting them pharmacologically would make a lot of sense. Or conversely, if we know they are not causal, let's stop trying to modify them. We need quantifiable traits, quantitative traits. This is important both so we can accelerate the science. So it's much easier to do a drug screen if your readout is lowering a biomarker or influencing kind of a linear trait. Sometimes there are radiographic quantitative traits. So we'll accelerate the research for drug screens, make it kind of clearer what animal models we should be focusing on. And then we need sorters or classifiers. We need to be able to easily, and in the ICU world, very rapidly sort our patients into these traits. So a trait that takes 2 weeks to come back is irrelevant in ICU medicine because we're making our decision in a matter of, minutes to hours, certainly not weeks. So can we look at some of the successes? And again, I'm not an asthma expert, so be kind to me here. But I keep going back to what remarkable success the field of asthma had with really intelligent use of molecular epidemiology embedded within randomized trials. So this example is from Prescott Woodruff's group. But I really just want to stress some of the details. So this was a randomized trial of inhaled corticosteroids for asthma patients compared to healthy controls. And it was a total of 42 patients with asthma and something like 28 controls. So what we would consider a pretty tiny study, but they used the power of randomization and very smartly, patients had to have a research bronchoscopy at entry and then after they're being randomized to fluticasone or control. So with this research bronch and bronchial biopsy, they did gene expression. And the first question was, how does the bronchial gene expression differ between asthma and controls? And they kind of came up with this first list of genes. Next, they asked, okay, if I take my randomized component and compared those treated with fluticasone to not, and here we actually had a paired, we knew your baseline before fluticasone, we could also generate a list of fluticasone mediated genes. And really interestingly, several of the genes were on both lists. So dysregulated in asthma and regulated by corticosteroids. And these three, periostin, serpin-B2, they then went on to show that they were both IL-13 regulated. And so now you have these candidate genes that might be drivers, and that you could actually use this gene expression to say, hey, we start to understand at least this part of this IL-13 driven asthma, and we have some readouts for it. So causal inputs are quantitative and could treat it. And of course, we're all pretty familiar now with our, I think I'm showing here our GINA guidelines where we step up in therapy, but we're pretty comfortable with treating patients with IL-13, IL-4, IL-5 targeted treatment for our most refractory asthma when it expresses some of these phenotypes. Our asthma colleagues didn't stop there. I think in another really impressive translational study, they looked to see if they could find a peripheral blood signature that would inform on corticosteroid response based on the gene signature that you would see in vitro by treating bronchial epithelial cells with corticosteroids. So now again, you might have a classifier that you could do much simpler without that bronchoscopy, which we all know is always a little risky in patients with asthma, not our favorite patients to bronch. And so now we could even move this one step further to peripheral blood. So again, things that they did so well, they leveraged a randomized controlled trial so they could be really confident about what was regulated by the therapy. They knew their tissue and could use gene expression. They used quantitative traits, IL-13 regulated genes, IgE, and they generated this classifier. Another example that might be a little further afield from our critical care expertise, but I think is so interesting from maybe a genetic epidemiology, is many of you might be aware that African ancestry is a huge increased risk for having certain types of chronic kidney disease, especially proteinuric kidney disease like FSGS. And genome-wide association studies have really definitively shown a very strong link to an area of the genome that's in which variation is private to African ancestry. So I'm showing you here a map. Anywhere you see kind of orange or red, that would be red homozygous or orange heterozygous for some of these variants in this APOL1 gene. But we really couldn't answer why. And so about a decade's worth of research between the GWAS and some of these studies showed that APOL1 is a potassium channel. And super interestingly, these mutations restore the ability of APOL1 protein to be lethal to the tsetse fly. That's what I'm trying to show you there. And so these variants may have been selected by tripos, I can't even say it, trypanosomiasis. I think I'm putting in extra. Trypanosoma brucei, right? African sleeping sickness. So that might explain why there's genetic variation in sub-Saharan Africa that we don't see elsewhere. And again, this was a crazy odds ratio increase of 29 for these types of kidney disease. Once they knew what these, that APOL1 is a potassium channel, then they could use this knowledge to say, okay, we can do drug screens to say what influences, how can we either turn on or turn off APOL1? And so they did drug screens to find a pretty potent APOL1 inhibitor. And then did a genotype guided precision medicine trial saying that if you carry one of these deleterious polymorphisms and you have proteinuria, we enroll you in our trial. And this drug had a remarkable effect on decreasing proteinuria and is now in phase 3 trials to try to see if it helps reverse or mitigate some of the chronic kidney disease. This is a very small subset of chronic kidney disease. And so this is an expensive drug. I don't know to what extent it's going to majorly impact health on a population scale, but it's also a really elegant way to show that we care about the health of all humans, including sub-Saharan variants that are very enriched in sub-Saharan Africa. And so now the next challenge will be, how do we get this drug to the patients who can benefit from it? I'll go a little faster. In cardiovascular disease, we all take it as truth that LDL is causal towards cardiovascular disease. And a little later in the talk, I'll show you some of the genetic evidence that helps prove that. But again, once you have this quantitative trait, great, figure out what regulates LDL, and you might have new candidates for therapy. So this is a Manhattan plot where we're literally, imagine we're laying out all the polymorphisms on every chromosome from chromosome 1 to 22, and then trying to plot the association with LDL as a quantitative trait. And each time it looks like you have a little skyscraper in your Manhattan plot, that would suggest a very strong association with multiple linkage disequilibrium partners. So I'm showing you here some of the genes that have been strongly implicated in LDL, but PCSK9 might be like a kind of very interesting story in that when these genetic screens were done, no one really knew what PCSK9 did. It was expressed in the liver, so why was it influencing LDL? And of course, research went on to show that this is what controls LDL receptor recycling. And so the density of your LDL receptors then influences your LDL serum concentration. And that helps clarify a therapeutic target, which again, now maybe these are early days, but there are now some CRISPR-Cas9 studies in non-human primates showing that one dose of this PCSK9 edited lipid nanoparticle could dramatically reduce your LDL by 75% in non-human primates. So not yet ready for prime time, but a really exciting way that we can translate some of this knowledge from genetics towards therapy. Again, I think some of the things that have helped facilitate these breakthroughs have been understanding these causal mechanisms and using genetics to help us do that, then honing in on those quantitative traits and trying to classify our patients. So why have we been limited in critical illness? What's kept us from doing this? Well, I don't want to complain, but it's hard. Which tissues should we be profiling? So I think of asthma, they biopsied airway epithelium. So what should we be biopsying for ARDS? And is it safe to do so? So maybe we all agree we should be biopsying the alveoli, but that's a very risky endeavor in ARDS. It might leave the patient with an air leak or pneumothorax, or it might deteriorate them. And our sickest patients are the ones who are going to be least likely to tolerate it. We use the blood or the BAL fluid, and maybe we're still a step away from kind of what's functionally dysregulated. In sepsis, I would love anyone to tell me what should we be profiling in sepsis. We really don't know. We look at the white blood cells, and I think there's a lot to be gained there. What about the site of infection? So we're never biopsying an infected kidney or colon. What about the pathogen side? So I think that's been a big challenge. Another challenge is that we can't always use some of the now widely available data, genetic data, on what regulates some of our traits. So for instance, we actually have a lot of knowledge about what regulates interleukin-6 levels at rest. But we ourselves, as humans, regulate IL-6 very differently during inflammation than we do when we're ambulatory. So we have this evoked trait problem during sepsis and ARDS, where we know that angiopoietin-2 is one of my favorite proteins that's a permeability factor. It would be very disadvantageous to always have leaky blood vessels. So we stringently control Ang2 during health, but maybe we need a little Ang2 to either grow new blood vessels or to recruit inflammatory cells to fight your pneumonia. So if we might need to study the genetics during the evoked stress. The same problem happens with imaging. So a lot of our imaging is very dynamic. If we think about, there have been some really elegant studies using quantitative radiographic traits. So for non-alcoholic steatohepatitis, which has recently changed names and I think now is MASH, not MASH, but we could use liver fat as a quantitative trait, and some genetic advances have been made, new candidates have been found. What's our quantitative trait in the lung if we're trying to image ARDS? Is it some degree of the consolidation, but what's here today might not be there tomorrow? And so is that genetically regulated? I think that's a real challenge, and maybe machine learning can actually help us solve that. But then we also have to think about our practice and how we use radiology, and we're not going to CT patients every day. We're not even X-raying them every day. Maybe we're going to ultrasound them every day, and is that a quantitative trait we could use? We need to think carefully about controls when we do critical care research. So again, our healthy controls, non-diseased ambulatory patients, is that the relevant group? Or do we need to do all our studies in patients who have the same risk to develop ARDS? So some of our work has used septic patients who do not develop ARDS as our control population. And this might be helpful when we have such kind of evoked stresses in critical illness. We need to have better EHR phenotypes. I think we've solved that for sepsis. We have yet to really solve it for ARDS. And then we need to leverage it on a large scale so that UK Biobank, you could go in and query ARDS. And we're getting better and better at clinical trials. I think COVID was such a turning point to really give us confidence that we can find mortality influencing treatments in our critical illness. Yet we didn't get a lot of samples we did randomized controls with samples, and we did molecular studies without randomization, but like we didn't match them up. And so we missed that opportunity to do what our asthma colleagues did with that really tiny 42 patient trial. And then of course, we need to be validating these markers so that we're using them in real time. Okay, so that was the downer part of today. Let's go on to what might we be doing well. So I'll be speaking, a lot of the work that we have done at Penn has been using a sepsis cohort that we call MESSI. I'll show you the abbreviation there. I am a soccer fan. So we initiated this in late 2008, and for some of the early career folks in the room, it's now over 4,000, but it started at 0. So it does take a long time, but it grows over time, and this has, I think, become a really powerful engine. So we enroll patients who are admitted to the ICU with sepsis as their primary diagnosis. If we think it's largely PE, but they're on antibiotics, we would not enroll, or largely DKA on antibiotics, we would not. But generally, we're looking for sepsis 3 criteria plus blood cultures and antibiotics. We collect blood on days 0, 2, and 7. We collect urine. We use the EHR, and then myself and a few colleagues read all the chest imaging, so both X-rays and CTs for the first, for day 0 through day 5, so the first 6 days after ICU admission, and we're phenotyping that to say consistent with ARDS, not consistent with ARDS, or equivocal. I can't tell. There's whole lung atelectasis. There's too much hardware in the picture. We also try to really maximize residual sample, and by that I mean clinical lab samples that would otherwise be discarded. So all your DNA we can get from your discarded CBC, and we can get that a week after, and it's still good DNA. So it can stay in the clinical lab while the clinicians think about add-on testing. We don't interfere with that, and then when that add-on window has ended, we can take it. We actually can use a lot of residual plasma. So at Penn, the citrated samples, the clinical routine is to spin upon receipt and definitely within 30 minutes. So this is fresh plasma. Citrate is the preservative. And then Penn's lab keeps it cold, which is not true everywhere, but they keep it cold. And then the add-on window is very short. It's really only 12 hours. So we're allowed to pick it up after 12 hours. And then we outplot it. We also, of course, do research blood sampling. Historically, we were doing that just for RNA, whole blood RNA, in a Paxine tube. We've since expanded, and now we're collecting peripheral blood mononuclear cells. Before we had the sepsis cohort, we actually had a trauma cohort that was started by Jason Christie, my mentor, and now chief of our division at Penn. He did this with Paul Lankin in a trauma population, thinking that with trauma, we really know when the insult is. And so it was a really nice kind of molecular epidemiology study. And in this trauma population, we had identified a variant in the gene interleukin-1 receptor antagonist. The gene name is IL-1RN, but it encodes the protein IL-1RA. That was associated with lower ARDS risk, lower sepsis mortality, and we validated that in David Christiani's large all-cause ARDS population. So we were pretty convinced we had a true genetic signal. And excitingly, this was an exonic but synonymous coding SNP. So it did not change protein sequence, but we did RNA sequencing to show that it seemed to be a site of allelic expression imbalance, meaning depending on which allele you had, if we looked at heterozygous patients, and it's let's say it's a kind of a CT polymorphism, you'd expect 50% of the transcripts to come from C and 50 to come from T if expression is equal. And we observed an imbalance where the C allele, the protective allele, was associated with 70% of the transcripts, suggesting that maybe there's something about you're more efficient at making IL-1RN transcripts, which should then translate into protein due to this polymorphism. And so, of course, that was really interesting because it raises the hypothesis that maybe this efficient IL-1RA production could be protective in sepsis and protect you against ARDS. And of course, for anyone my age or older, that's a little retro because there were many trials in the 90s. Steve Opel at Brown was the PI of some of these trials, but he describes drug companies and the New York Times calling him to say, is this drug gonna end sepsis? Will it end sepsis as we know it? There was a lot of hope for this. But in 2 large randomized phase 3 trials, the drug had a small and not statistically significant reduction in mortality. It didn't seem very dose responsive compared to placebo. So unfortunately, no mortality benefit. But we thought in light of our data, would the effect of this drug be uniform if we took into account some aspect of either your genetics or your expression of IL-1RA or IL-1 beta? And so the way you should do this, I'll refer everybody, the epidemiologists, to Kent and Rothwell, publish a lot about what's a proper way to do subgroup analysis in clinical trials. It's not appropriate to go and just look at all your subgroups and find a p-value. So like that's not recommended. But if you have a strong hypothesis that it should differ, the first thing you should do is ask yourself, is the effect heterogeneous? You're testing for effect modification, or said another way, a statistical interaction. So we asked whether the mortality effect of the drug interacted with your baseline IL-1RA level in these old clinical trial samples that Steve Opel had in his freezer, over 20 years old, and it's a cytokine, so that's all kind of a grain of salt. But we did observe interaction, and maybe shown a better way, among the IL-1RA high group, and we looked at high to find many different ways, whether we kept it by decile, or we dichotomized at the median, or dichotomized at what's called a Youden cutoff that optimizes sensitivity versus specificity. Whichever way we looked, we found that the IL-1RA high patients benefited from more exogenous IL-1RA, which was counter to our kind of simplistic thought. We thought it's the low IL-1RA patients who need more IL-1RA. That was statistically significant reduction in mortality compared to placebo, compared to the low baseline IL-1RA group, who, if anything, did worse when given IL-1RA. So I think how I put this together is that your plasma IL-1RA, we know it's first strongly correlated with your plasma IL-1 beta. And I think it may just be a tattoo that you have IL-1 dysregulation. And in some ways, the plasma IL-1RA, maybe it's less regulated than IL-1 beta. That would make sense because beta does bad things in parts of your body. So it's just a better marker that your IL-1 axis is dysregulated. So potentially, could this be a putative treatable trait? I've hopefully shown that it's relevant. It associates with mortality. It's measurable. It's pretty easy to quantify a protein. And IL-1RA is measurable in all of us. It's detectable even in ambulatory patients, and then, of course, gets much higher in sepsis. Is it treatable? The newest form of recombinant IL-1RA, anakinra, remains available. We use it for refractory arthritis. And during COVID, I'm showing you here data from the SAVE-MORE trial. So a group led by some of the Greek critical care intensivists tested anakinra in a moderately ill COVID population. These were hospitalized patients, but very few needed high flow or mechanical ventilation. So a little different than the patients that I think you and I cared for during that first spring wave and then Delta. but it had a mortality benefit. So there are attempts to potentially try anakinra again in a larger population. I mentioned angiopoietin-2 is near and dear to my heart. So our same early genetic studies in our trauma population, we found increased risk for ARDS with some variants in the angiopoietin-2 gene. And this was really interesting because others had already shown strongly that plasma Ang2, the gene product of our gene, strongly associates with ARDS risk among a sepsis group and with ARDS mortality. And so I'm showing you some data here from our MESI population. It's for each log increase in Ang2, your risk for ARDS is up 1.6, really strong. It's moderately genetically regulated at rest. So from the Framingham data, we have heritability estimates of around 27%, which is modest. But our question was really, what about evoked angiopoietin-2? Because we know Ang2 is pretty low in you or me while we're walking around. And so we did what's called a quantitative trait locus analysis, where we first asked, what's the relationship between the genetic Ang2 gene variation there and plasma Ang2 during sepsis? And we could kind of locate that peak genetic association. And when we overlaid it with our trauma GWAS, this is a different population, now trauma looking with ARDS, it really lined up nicely that these were linkage disequilibrium partners. So this led us to think that we could apply a technique called Mendelian randomization. That's a really fancy name to say an instrumental variable analysis. So an instrumental variable means that if I think I have either a variable that's hard to measure or that has risk for confounding, if I have an instrument, in this case our instrument is genotype, where I could predict a very pure portion of the variability in that variable. So like genetics assigned at birth, not affected by your, we don't think not largely affected by your environment. And so if we could define the genetic variation in evoked Ang2, and if that genetic variation associates with outcome, that's strong evidence that we may have a causal relationship. And so this is kind of a tool to infer causality. If we go back to the LDL example, these are some of the experiments that were really helpful to prove that LDL is causal for coronary disease, and that markers like C-reactive protein are not causal for coronary arterial disease. If we have in this case, a SNP in the LDL receptor that's strongly associated with or moderates a good proportion of LDL, then we can also ask what's the relationship between SNP and coronary disease and see that this was a negative association. So that's why the odds ratio is less than 1. And in a simplistic way, if you think about this, if we adjust in this relationship between LDL, SNP, and coronary disease, if we adjust for LDL, there is no further relationship between SNP and outcome because it's all mediated by the change in LDL. So we were asking, could we do the same with plasma angiopoietin-2? And again, here's kind of our simplistic framework. And a lot of this work was done by my colleague, John Riley at Penn. So if we took our top SNP that was strongly associated with plasma Ang2, and then we knew plasma Ang2 was strongly associated with ARDS risk, we were really excited to see we did have an association between SNP and ARDS. But this is what we would call a weak instrument because our SNP explained a very tiny portion of risk. And really to do an instrumental variable, you'd like to explain about 10% of the variation in your variable. So if we used multiple SNPs in the Ang2 gene, we could explain closer to about 8%, still not perfect. But that allowed us to kind of come up with a genetically determined portion of your Ang2. And that was strongly associated with ARDS, even accounting for covariates like Apache, whether or not you had pneumonia, which strongly influences your ARDS risk, and of course, genetic ancestry. So to do the same, we think we have a relevant trait. It's measurable. Plasma Ang2 is measurable. Is it treatable? Soon, I think we will be able to say yes. So there are a lot of drugs in development. This Ang2 and its receptor Tie2 is of great interest to oncologists because the thought about most metastases is that you need neoangiogenesis to metastasize. We might need a slightly different drug, right? We want short half-life, not something that we could dose once a month. But there are some of these drugs. I think that razuprotafib may have failed in early trials, but I think vasculotide is one that might be progressing. And so I'm excited to see if this comes to bear. And of course, there are other ARDS quantitative traits, putative traits. Tiffany Jones, kind of at Penn, has shown something very similar with soluble RAGE. David Christiani's group have done a lot of work on both platelet counts and platelet trajectory. And again, I think as we get these quantitative traits, then we can start using the EHR and kind of larger cohorts, maybe even the APS cohort, eventually to try to better understand the genetics of these evoked traits. Okay, so let's switch a little bit to some of the classifiers. We know gene expression has been so powerful in asthma. Have we been able to leverage this in sepsis or ARDS? And most of this next part will focus on sepsis, where I think we all believe that whole blood, which when we extract RNA from whole blood, really we're looking at your white blood cells RNA. So a group led by Julia Knight and Emma Davenport at, I think this was Oxford, did large-scale gene expression in some pretty critically ill pneumonia populations. And when they looked at the most variable genes and just those variable genes and plotted those as principal components, they said it really seems like we have two groups here that they named SRS1 and SRS2 kind of in the blue and red. And in two populations, they were able to show that SRS1 was a high mortality group. Again, differed by how sick the population was at baseline, but SRS1, high mortality, SRS2, maybe a more favorable or regulated host response to sepsis, where these patients had fairly low mortality. And potentially even more interesting, when another group from the UK led by Tony Gordon and David Ancliffe looked retrospectively within the VANISH trial. So this was, I think, a factorial trial in sepsis where one factor was randomizing you to sepsis, to steroids, no steroids, and the other was to vasopressin as part of your pressor regimen. They found no effect in the overall population, but when they looked at a subgroup in whom they could test SRS, it looked like the SRS2 patients, who I'll remind you were the low mortality patients, these are supposed to be the survivors, seem to be harmed by steroids dramatically, like huge difference. This is so interesting. It is still preliminary. This is a very small subgroup. So I kind of put up here, the subgroup is only 55 patients, but it's really food for thought and does show that that effect modification, that maybe we need to know what your host response is before we try to modify it with corticosteroids. So SRS could definitely be a treatable trait, and you know, the treatable portion here, it might be a do not treat if SRS2. The same group, Emma Davenport's group, have now gone on and tried to make this even more user-friendly by making it a quantitative trait, not just a yes, no, are you SRS1 or SRS2? And you can now do this with either microarray data or with RNA sequencing from whole blood. They've shown that it reliably associates with kind of poor outcomes. And I think it's giving us some clues to mechanism as well. And some of the features that they're seeing, including neutrophils that tend to degranulate easily, immature neutrophils, and this signal for what's called emergency myelopoiesis, making too many early neutrophils, maybe at the cost of having lymphocytes and having this kind of balanced immunity. Something we saw a lot during COVID, where we started paying attention to the neutrophil lymphocyte ratio, but which is true in most types of sepsis. We see a lot of those same features when this is some work done by Heather Giannini, who was an early career faculty with me at Penn, when she looked at gene expression to just say, could we discriminate sepsis survivors from non-survivors? A lot of these same features arise. I think time will tell very shortly whether this is a treatable trait. And as we think about some of our COVID learnings and baricitinib, which is a STAT inhibitor, it very well might have been modifying something in this emergency myelopoiesis. So I think this is an exciting thing. Are we going to use SRSQ as our quantitative trait? Maybe. We'll need some tools that are a little more facile than doing RNA-seq from the blood. That's not a minutes to hours test. But I think there are ways to adapt RNA to be rapid diagnostics. And so maybe we could get answers on those pretty quickly. You're all probably quite familiar with classifier successes in ARDS. I almost feel like I don't need to say too much about this because Carolyn Calfee and Michael Maffei have done such an amazing job of teaching us about this. But very consistently in clinical trial populations as well as observational cohorts, Carolyn has applied latent class analysis, saying like, just put all the data, put all the clinical variables, the ventilator variables, and some biologic variables, plasma biomarkers, and let the computer tell me how many groups do I have. And very consistently, she's found that we have two groups that she termed hyperinflammatory, because what was most different about them were very high plasma IL-6, IL-8, TNFR1, and relatively low bicarbonate systolic blood pressure protein C. So the biomarker seemed the most discriminatory. Bless you. Another group in the Netherlands found very similar things with a slightly different method. So is hyperinflammatory ARDS a treatable trait? Probably. So Carolyn has shown retrospectively in at least 4 trials a heterogeneous treatment effect, including with Pratik Sinha, they looked at activated protein C, Zygris. That was very in phase in the University of Chicago in the early 2000s, but shown a differential effect. So maybe this is something we really need to know. And there are some clinical trials and platforms really focused on, can we get the knowledge to infer your hyperinflammatory type quickly at the bedside? And there are some platforms to do rapid proteomics. There's something called Randox, there's something called Ella, where we can get your ELISA in 90 minutes. And that's probably fast enough for us to make a decision about a drug. So I think that will be what we see in the future. And I'll close just with a little bit of maybe some classifier success that our group had during COVID, where the one thing we added to MESI was to do flow cytometry. And I think I was speaking with a few of you about the COVID experience. I think Philadelphia's experience was somewhat similar to New Haven's in that we were very fortunate to see what was happening in New York, but have that week or two to prepare before it was completely overwhelming. And of course, I think we used that to, ready our ICUs, ready our workforce. There was so much uncertainty at the time, but it also allowed us to ready some of our research infrastructure. And so how Penn chose to do that was because our group were already running a cohort study of sepsis we just adapted that to, it was COVID sepsis. PPE was, of course, very limited, so it was physicians who were doing the consenting rather than coordinators. And then I was incredibly fortunate to partner with some of our outstanding immunologists to do some of this flow cytometry work. So first, I thought I would just show you the population. So when all was said and done, in about 18 months, we had enrolled a little over 400 patients. And I highlight here some of the demographics. So 60% of our patients were Black, about 33% white. And compared to our, where we had cut the data, our messy kind of historical control, this was a total flip of our demographics compared to what we would typically see from our ICU. And I can tell you as a clinician and as speaking with a lot of these patients and their families, because I was calling them to enroll, if I wasn't seeing the patient was ventilated. So many were from 3 zip codes right around Philadelphia, very disadvantaged neighborhoods. And, you know, when you think back to the being told to socially isolate, you need some means to socially isolate. You need to not have to take the bus. You need to live in a small family or, you know, a few posse person home. So it doesn't work when you have multi-generational homes. So it was really quite striking. And so I think this was quite representative of who we saw. A higher proportion of ARDS if you went by our new global definition. But a lot of these patients were non-ventilated ARDS or non-intubated ARDS. And thankfully, a slightly lower mortality than we typically see from our largely bacterial sepsis. We started working with Mike Betts, an immunologist, and his team, first to just understand what did the whole blood look like, what were the leukocyte populations. And what I'm showing you here are Tisney plots where like clusters with like, and then we can use informatics to kind of figure out kind of with these flow cytometry markers, kind of what these are. So compared to HC as healthy donors, the severe COVID group, first you notice this huge expansion of black dots. Those are neutrophils. So this is a very neutrophilic disease, which you recognize. And if we informatically remove the neutrophils that look at the remaining mononuclear cells, I think you can appreciate other differences, again, between healthy donors and COVID, where maybe purple, these are B cells, contract. T cells here in orange compared to here, again, contract or lower. And you see the emergence of this new population in red that we didn't see at all in healthy donors. The plasmablasts, yeah, this really struck us early. So plasmablasts are antibody-producing cells, kind of an early B cell. And the proportion of plasmablasts in some patients was 50% of their B cells. This is a marker of severity that we usually see in severe viral illnesses like dengue or Ebola. Usually we only see it in the hours after a vaccination, and then it's quite low again. So that was quite striking. And by using, we then went on, this is work with John Ware's group, where we did very high dimensional flow cytometry and focused especially on these kind of depleted T cells, but tried to really come up with integrating all these different features. And I think some of the things we learned were that it's quite heterogeneous, even among hospitalized severe COVID patients. We were enrolling both from the floor and from the ICU because ICU meant different things during COVID. We didn't think floor location was a great tell of the patient's severity. But we saw these T cells and B cells, though they were low in number, they were also different. And some of them were highly activated. If we used markers like this HLA-DR positive CD38, they were active, they were proliferating, and maybe exhausted. They were also expressing high degree of PD-1s. So if we put those all together and compared them to patients who were never exposed to COVID or had recovered from COVID, we actually had a couple patients who were never hospitalized, but experienced COVID early and gave us blood. It definitely saw that we could discriminate, But then there was heterogeneity even among the hospitalized patients. And I'm not sure how well it projects, but hopefully you can appreciate there's some very dark red or maroon and then kind of this lighter pink. So that was our WHO ordinal scale, with the darker pink being the more severe intubated patients. And while that severity seems pretty uniform throughout the second principal component, it did seem to cluster. There was a higher proportion of them kind of further along the x-axis. So we kind of integrated this as a quantitative trait that we called your immunotype, where it seems like patients kind of bucketed into 3 types, where immunotype 1 was the highest severity associated with ataxia and mortality, really highly activated CD4 and CD8 cells, that high plasmablast response, and kind of altered, none of the T cells looked normal. Whereas immunotype 2 had some of the things you'd expect to be happening as you're fighting virus, like emergence of memory B cells or proliferating B cells. So maybe this immunotype 2 was a more balanced response. We had a low number of inpatients. Let's see, I can actually go back. I think there were 3 hospitalized COVID patients who actually clustered with healthies. One of those I remember really well. He was admitted early for intoxication, and he didn't have a home to go to. So we felt a little stuck, like, what do we do with this man who's febrile and has COVID? So he was very happy to participate in research, but I think it made sense that he was immunotype 3. 2 others, though, had clinical pneumonia and required oxygen, but still had this very bland kind of non-immunotype response. So I'll just stop here saying, like, could this be a new treatable trait? We've been able to replicate certain features of that high immunotype, the T cell activation. Plasmablasts, actually, your proportion of plasmablasts do not well predict your severity. Probably more where you are in the time course since your infection. But there's a very reliable qualification process our oncologists use all the time to take a flow marker and make that a clinical test. So if we find these, this could be a clinical test. And again, the question would be, is it timely enough to be helpful for critical illness? And then, of course, what we'd really like to do is test the response to therapy, which we started to do a little with the ISI COVID trial, and I know Yale was also a site. But I think the shifts in the pandemic, we just didn't really have enough patients to make firm conclusions. So I think I'll just end there by saying, I think I do believe, especially today, like I think maybe we all need to go back and watch Ted Lasso. But I do think we can accelerate future success by finding some of these quantitative traits, using that knowledge to find those driver mutations and classify, and then hopefully start leveraging our randomized trials. And with that, many thanks. This type of work always takes a village.
Thank you, Nuala. That was a fantastic talk. We have time for a couple of questions. Just so you are. Can I find if that's not working? And for our Zoom participants, if you could put your questions into the chat, that would be great. Thank you.
Thank you so much. Great talk. I think in addition to obviously some of the challenges that you highlighted, right? A lot of these studies have the challenge that some of the data that we have collected is cross-sectional rather than accounting for the dynamics. And I think that was one of the criticisms for the studies in the 1990s, right? In terms of like when you should administer some of these immunomodulators, right?
Yeah.
So, what are your thoughts and how are you accounting for that in some of the current studies?
Yeah, great question. So again, the question is, how do we account for the dynamic nature of anything we're measuring during critical illness? And we know, like a cross-sectional study, can we really rely on? It's something we definitely haven't solved. I think I think there's a lot of utility to ICU admission. Something brings patients to our attention. And so I think that's a great place to start. And of course, we often do not have a pre-sample for pre-critical illness. Some ways our team has thought about using is, could we use a recovery sample to estimate pre, knowing that that's not perfect, like maybe they're forever dysregulated. But for at least some of the cytokines, you would expect that you're going to get closer to normal. And so we're trying to do that a little bit with both gene expression and some plasma protein work. Of course, we do like the longitudinal samples and trying to understand trajectories. We know some of that historically, that usually ICU admission is the peak for most patients and most markers will decline over time. But what we don't know, we haven't done the classifiers repetitively. I think Hector Wong did some of the gene expression classifiers and found that it changes a lot between day 0 and day 2 in children with sepsis. So those gene expression classifiers may be quite dynamic. And maybe there's information in that, if it's getting closer to normal, does that tell us about your trajectory is improving? But I hopefully, to plug the APS a little bit, the ARDS pneumonia and sepsis study will be getting multiple time points. I think day 0, 2, possibly 4, 7, and 14, and then again at 3, 6, and 12 months. So maybe that will be a resource to the community to do some of that work.
Thank you so much. Really impressive what you've been able to accomplish despite the long list of barriers that you started your talk with. I was particularly interested in the ANG2 work, and you indicated you were very interested in that. And if I understood you correctly, it causes both sort of capillary leak and a link to coronary outcomes or cardiovascular outcomes. Is that?
This one, ANG2, I think I'm not aware of it being a cardiovascular risk factor. I think ABO is for things like clotting. Not to my knowledge. It was tested in Framingham just as a large molecular epidemiology. So from Framingham, we know we can estimate its heritability, which is kind of modest at 27%. Things like lipids are probably well over 50% in heritability.
No, the reason I ask is in the field of sleep medicine, where I work, there has been some really interesting data about high CPAP pressures and elevation of ANG2. People are concerned
that would concern me.
Yes, and it was initially discovered actually in a cohort of patients who have sleep apnea and interstitial lung disease, so potentially high airway pressure. So I'm wondering if there may be something related to high airway pressures, that substance in ARDS that could be.
Oh, that's so interesting.
Not just the ARDS, but the pressures that the lungs are under.
It can be almost like a mechanosensor.
Yeah.
And I would imagine that these concentrations you're seeing are different than ambulatory, but probably still orders of magnitude lower than during sepsis, but concerning nonetheless that if your lung is sensing enough stretch to leak, we do know from, if you put ANG2 on an endothelial monolayer, it becomes leaky. So it's definitely an injury marker. So maybe that would be something to factor in as we're titrating our pressure settings. I didn't know that.
Thank you, Nuala. So as you're hinting at, the diagnosis of sepsis and ARDS and pneumonia are all really messy, right? Yes. Literally messy.
Yes.
And we just had Barbara Jones here talking about the diagnosis of pneumonia.
Yeah.
And it occurred to me that we're sort of taking as a given that we have a population that meets clinical criteria for a diagnosis. And then we're following up with what sounds like pretty precise markers of something that's going on. And I'm wondering if you're looking ahead to thinking that maybe it's not just an issue of looking at subtypes of a diagnosis, but the actual diagnosis itself, right? Because so much is wrapped up in things like clinical decisions, like like I intubate and Clar doesn't, right? Or I start this pressor and Lauren starts another.
Yes.
And so I'm wondering where you see this going in that direction.
I think that's such a great question about what about the diagnostic side of these syndromes? So can we get more precise? And I think you're hinting at, to some extent, it's the pathogen side. So how confident are we that we do or do not have a pathogen? There are many tests available that aren't always used, and Penn does not use BioFire, but I did hear we just will start using a molecular diagnostic for cultures that have already started to grow GNR. So it'll be a more rapid identification of GNR. But it's too expensive right now to say we're going to use a molecular test every time we order a blood culture. But maybe that's where things are going, to get at least at the pathogen. So we could look at the host response and we could look at the pathogen side. And the APS is collecting a lot of microbiome data, so we may be able to answer some of that more precisely. We still have that issue with when do we intubate, when do we start high flow, when do we start pressor. Those are some of the eligibility criteria for APS, so we will not solve that problem with APS. Though I do know that some of the group are very interested in trying to do target trial emulation by just collecting as much data about the interventions that we are performing while we're doing this molecular phenotyping, so that we can at least try to estimate the effect some of our treatments might have, and be able to phenotype patients at baseline. Even though we didn't know it, like retroactively, we can say, oh, look, this was a high Ang2 person who got lots of fluid and not much pressor. And is that a harmful strategy compared to, if the Ang2 is high, maybe more fluid is going to be harmful.
Oh, there we go. Lauren warned me it took a little while to turn on. A great talk. I actually had a follow-up question to Clar's, but that wasn't my primary question. We actually had a cohort here where we looked at Ang2 and sepsis. And so what would be interesting to look at that population is that the severity of illness as well as the hemodynamic derangement drove the angiopoietin elevations in serum. We were looking at serum and not plasma. And you can imagine somebody who has bad sleep apnea that's maybe partially undertreated. They may have more vascular pressure swings in the setting of their sleep apnea, and it may not be the pressure itself, but I don't know. My question I was going to ask was, in our cohort, the thing that was a little hard to tease out was almost all patients that we see who have ARDS had sepsis. And so how do you separate out the sepsis piece from the ARDS itself? Because it seemed like the hemodynamic perturbation was a driver of the angiopoietin, but it's obviously multifactorial. So just question how you think about that.
Yeah, great question. Again, can we separate what's ARDS and what component is attributable to the sepsis or septic shock? I agree with you that I think Ang2 is a great marker for shock. And so shock is also a risk factor for ARDS, and it becomes a bit confounded. Certainly there is one, I want to say Gallagher is the last author on one paper that's now 15 years old, where she compared sepsis to sepsis with hypoxic respiratory failure, and the hypoxic respiratory failure did have higher Ang2 than just sepsis, but they were sicker patients, so probably a higher proportion of shock as well. And does it matter? So like maybe APS will tell us that if the trait is Ang2, regardless of how you're expressing Ang2, the treatment is anti-Ang2. And if we know it's safe, like there are lots of ifs here, I'm not recommending we start that treatment today. But again, and Richard and I spoke about, I think we also have to open ourselves to the thought that probably it's not going to be one drug, that probably as we define these traits, you could be trait positive for 2 and trait negative for 4, and you get the combo for the 2 you have, as long as those will work well together. And I think if we get comfortable with that, we need to think about that in the experimental phase, so that in animal models, we're testing these combos and we don't find out that one, I think of our remdesivir Plaquenil experiment during COVID where we were giving remdesivir and inactivating it with hydroxychloroquine. Like that was silly, but that's exactly what we did. So like I think as we at least keep an open mind to that, maybe we would be prepared. So we don't figure those things out in people, but we figure those out in safer models.
Do we have any questions in the chat? No? All right. Well, thank you everyone for attending and